Welcome to G Fun Facts Online. The pill you take for a headache. The injection that fights off a dangerous infection. These are marvels of modern science. They represent hope and healing. But what happens when that trust is broken? What happens when the medicine designed to save you becomes a poison? Today, we're diving into the unseen world of pharmaceutical contamination, a story of microscopic invaders, molecular stowaways, and the high-stakes scientific detective work that uncovers the truth behind a drug recall. A drug recall is the ultimate emergency brake for the pharmaceutical industry. It's a massive, urgent effort to pull a potentially harmful product from shelves and medicine cabinets to protect the public. While recalls can happen for simple reasons like a typo on a label, the most terrifying ones stem from contamination, the accidental presence of a foreign substance, and the journey to figure out how a trusted remedy turned toxic is a masterclass in forensic science. First, let's meet the culprits. In the world of pharma, there's a whole rogues gallery of potential contaminants, and they fall into three main categories. The first are physical contaminants. Imagine finding a tiny shard of glass, a metal filing, or a stray fiber in your medication. These are unwanted particles that sneak in during manufacturing, perhaps from poorly maintained equipment, or even from the clothing of factory workers. While they might seem minor, for an injectable drug, a single particle can be deadly. It can travel through your bloodstream and cause a blockage, called an embolism, or trigger a severe inflammatory reaction. Next, we have the chemical contaminants, the molecular intruders. These are unwanted chemicals that can be introduced at any point in the manufacturing process. It could be a solvent used to synthesize the drug that wasn't fully removed, or even a cleaning agent left behind in the machinery. One of the most dangerous forms is cross-contamination, where trace amounts of one powerful drug accidentally mix into a batch of another. But a particularly infamous group of chemical contaminants is nitrosamines. You may have heard of these in the news, linked to recalls of common blood pressure and heartburn medications. Based on animal studies, they are classified as probable human carcinogens. This means that long-term exposure, even to tiny amounts, could increase the risk of cancer. They can form unexpectedly during the chemical reactions of drug manufacturing, and their discovery has triggered a massive global effort to hunt them down and eliminate them from our drug supply. Finally, and perhaps most alarmingly, we have microbial contaminants. These are the living threats: bacteria, viruses, and fungi. They can come from contaminated raw materials, impure water, or even from the people making the drugs. For sterile products like injections, the presence of a single living microbe can be catastrophic. It can lead to severe infections, sepsis, and death. And even if the microbes are killed during sterilization, they can leave behind toxic garbage. A class of bacteria known as gram-negative bacteria, for instance, leaves behind a substance called an endotoxin. If even a minuscule amount of endotoxin is injected into your bloodstream, it can trigger a massive immune response, causing high fever, a sudden drop in blood pressure, and potentially fatal septic shock. So when a contaminant is found, how do scientists figure out what it is and where it came from? This is where the detective story really begins, in the high-tech world of analytical chemistry. The first step is to separate the bad guy from the good guy, the contaminant from the drug itself. The go-to tool for this is called chromatography. Think of it like a molecular racetrack. A liquid or gas containing the sample is pushed through a long column. Different molecules have different chemical properties, causing them to race through the column at different speeds. This allows scientists to isolate the contaminant, getting a pure sample to work with. The two workhorses are high-performance liquid chromatography, or HPLC, and gas chromatography, or GC. Once the contaminant is isolated, the next question is, what is it? This is where spectroscopy comes in. Mass spectrometry, or MS, is like a hyper-precise scale for molecules. It zaps the contaminant, turning it into charged particles, and then measures its mass, This gives a huge clue to its identity. Another powerhouse tool is nuclear magnetic resonance, or NMR spectroscopy. It uses powerful magnets to probe the structure of the molecule, giving scientists a detailed blueprint of how its atoms are connected. The real magic happens when you combine these techniques. Scientists use what are called hyphenated techniques like liquid chromatography mass spectrometry, or LCMS. This brilliant setup combines the racetrack and the scale into one machine. It separates all the components in a mixture and immediately weighs each one, allowing scientists to detect and identify impurities at incredibly low levels. We're talking parts per billion, which is like finding one specific blade of grass in 1,000 football fields. With this chemical fingerprint of the contaminant, the forensic investigation moves from the lab to the factory floor. The identity of the substance provides the crucial clues. If the contaminant is a known byproduct of a specific chemical reaction, investigators know to look at the synthesis steps. If it's a cleaning agent, they scrutinize sanitation procedures. If it's another drug, they review production schedules to find where cross-contamination could have occurred. This meticulous process uncovers the how and why, so it can be prevented from ever happening again. The reason this all matters so much is the devastating human cost. When it comes to biologic drugs, these are the large, complex therapeutic proteins like monoclonal antibodies. A unique problem arises: protein aggregation. These giant molecules are delicate. If they get too warm or are shaken too much, they can unfold and clump together. These clumps, or aggregates, aren't just useless. They can be dangerous. Your immune system can see these clumps as a foreign invader and launch an attack, creating anti-drug antibodies. At best, this neutralizes the medicine, making it ineffective. At worst, it can cause severe allergic reactions or even trigger an autoimmune condition where your body starts attacking its own healthy proteins. To understand the real-world stakes, let's look at two landmark cases that changed everything. The first is the New England Compounding Center, or NECC, tragedy of 2012. It began when a devastating outbreak of fungal meningitis started sweeping across the United States. Patients were getting sick and dying after receiving steroid injections for back pain. The investigation, led by the CDC and FDA, traced the source to vials of a steroid produced by NECC, a large-scale compounding pharmacy. The vials were contaminated with several species of mold, most notably a black mold called Exorohilum, which is normally found in soil and on plants. This fungus isn't typically harmful to humans, but when it was injected directly into patients' spinal canals, it was catastrophic. The outbreak ultimately affected nearly 800 people and caused 64 deaths. When inspectors raided the NECC facility, they found a complete and shocking breakdown of sterile manufacturing practices. There was visible fungal contamination in the so-called clean rooms. Equipment wasn't being properly sterilized, and the company was shipping out huge batches of injections without even waiting for the results of sterility testing. The tragedy exposed a dangerous regulatory loophole, and in response, the U.S. Congress passed the Drug Quality and Security Act, giving the FDA much stronger oversight over these large-scale compounding facilities. The second case is the heparin crisis of 2008. Heparin is a widely used blood thinner derived from pig intestines. In 2007 and 2008, doctors began reporting a wave of severe adverse reactions in patients receiving heparin, sudden drops in blood pressure, and life-threatening allergic responses. An international investigation was launched. What scientists discovered was shocking. The heparin hadn't just been accidentally contaminated, it had been intentionally adulterated. Someone, somewhere in the global supply chain, had cut the expensive active ingredient with a cheaper, chemically modified substance called oversulfated chondroitin sulfate, or OSCS. This imposter molecule was cleverly designed to fool the standard quality control tests of the time. To solve the case, scientists had to invent new, more sophisticated analytical methods, using advanced NMR and other techniques to finally unmask the contaminant. Pharmacologists then discovered that OSCS wasn't inert. It was actively triggering the dangerous inflammatory pathways that were making patients so sick. The crisis led to at least 81 deaths in the US alone and exposed massive vulnerabilities in the global drug supply chain. In response, regulators worldwide implemented much stricter testing requirements for heparin, now including the advanced techniques developed during the crisis to screen for OSCS. These horror stories are powerful reminders of what can go wrong. But the ultimate goal isn't just to be good at responding to crises. It's to prevent them from ever happening. This is achieved through a multi-layered shield of prevention. The foundation is a set of rules known as Current Good Manufacturing Practices, or GMP. Enforced by regulators like the FDA, GMP is the rulebook for how to make drugs safely and consistently. It covers everything from the design of the factory and the quality of raw materials to the training and personal hygiene of staff. More recently, the industry has been adopting a smarter, more proactive philosophy called Quality by Design, or QBD. The old way was to make a huge batch of a drug and then test a few samples at the end, hoping everything was okay. Quality by Design flips that idea on its head. It's about building quality into the product from the very beginning. It involves deeply understanding the entire process, identifying all potential risks, including sources of contamination, and designing controls to mitigate them every step of the way. This is all managed through a robust quality management system. This includes rigorously vetting and auditing suppliers of raw materials, designing facilities with advanced air filtration and clean rooms, validating every single cleaning procedure to prove no residue is left behind, and extensive training for all personnel on how to work in a sterile environment. And because the drug supply chain is now global, an active ingredient might be made in India, formulated into a pill in Germany, and sold in the United States. International cooperation is essential. Organizations like the International Council for Harmonization bring regulators and industry together to create a single high standard for quality and safety that applies everywhere. The science behind drug recalls is a story of a never-ending vigil. It's a silent war fought in laboratories and cleanrooms against an unseen enemy. The work of analytical chemists, pharmacologists, and regulatory scientists is a critical, often invisible shield that protects public health. The tragedies of NECC and the heparin crisis serve as stark reminders of the constant threat and the devastating consequences of failure. But they also sparked progress, driving new technology and stronger regulations. The journey from a contaminated vial to a global recall is a complex and urgent one, but it's a process guided by science and driven by an unwavering commitment to ensure that the medicines we depend on are, and always will be, worthy of our trust.